Continuing our discussion on vision, I want to talk about receptive fields. The finding that information doesn't go straight from the rods and cones to the brain in sort of a cable. In fact, information processing is taking place all along that route. Every time there's a synapse, there's an opportunity for information to be processed. So the retina and the transmission to the visual cortex involves complex processing of visual stimuli to produce contrast effects, detections of edges, detection of angles. We have a lot of information when it reaches the inner layers of the visual cortex. If you look at this, it's simply a bar of gray, and it's even from the left side to the right side. But if I put it into contrast with other bars of gray, from very dark to very light, we see mock bands. We don't see an even band from left to right, but on the left side of every band, it looks brighter, and on the right side of every band, it looks darker. In other words, the processing that takes place in the optic nerve and in the thalamus actually produces contrast effects so we can better see edges, better see lines, and that's what we want to talk about today. In fact, the whole idea of receptive fields was brought forth by David Hubel and Torsten Wiesel at Harvard, and they won the Nobel Prize in the early 80s, along with Roger Sperry that we talked about when we talked about split brain research. The three of them won the Nobel Prize, and Hubel and Wiesel won it for their findings on receptive fields, the finding that information processing takes place early in the visual system as it goes towards the cortex. They studied cats, and they put microelectrodes in the brain of the cats so that it was firing on one single neuron. In other words, a single cell, and then what they would do is flash a light in the screen in front of the cat's eyes, and they would look to see what happens to that neuron as the light stimulated the eye. And what they found was very interesting. If they were looking at very early in the optic nerve, where it reaches the thalamus and sort of the ganglion cells, which we know come from the rods and cones, the bipolar cells, the ganglion cells, they found that the light on the screen did some very interesting things. If there was no light on the screen, they did get firing, sort of spontaneous firing that occurs in the cells without any stimulus at all. If they place a positive stimulus, that is a light, I have it as a dark dot, so think of that as a light on the screen, then it actually increased the firing of that single cell. So light, increased firing, no light, decreased firing. And then they found if they go outside of that center area into the periphery of a area on the screen, they actually got reduced firing from the spontaneous firing when there was no light at all. And then if they went outside of this area on the screen, they got firing like they had when there was no light at all. So it seems that we have a receptive field, an area of the visual field that will stimulate this single cell. And if it's in the center of that receptive field, they get positive increase in firing. And if it's in the periphery of that receptive field, they got decreases in firing. In fact, they found two kinds of receptive fields. One where the increase in firing occurred in the center of the receptive field. Then there were other receptive fields which the center produced inhibition of firing, and the periphery of that receptive field produced increases in firing. So there was an on-center cell and off-center cells. These round receptive fields that are stimulated when the light is located in the center of it or in the periphery of it, depending on what type of cell it was. So light on the screen depends upon these round receptive fields that either have an on-center or an off-center in their description. Now, if you line these cells up, these receptive fields up, through synapses, so that the output reflects this kind of receptive field, sort of a rectangular field with an on or off center and an on and off periphery, which occurs later in the visual pathway, going up to the inner layers of the cortex. In other words, if a light was right in the middle of that, we got stimulation, increases in firing, and if it's in the periphery, we got decreases in firing. So if the light actually moved across the visual field, the receptive field, we can actually detect edges or lines. And then if we combine those edges and lines together, higher up in the visual pathway, they found angle detectors. So if a right angle moved through the visual field, the receptive field actually was firing. There were also receptive fields for other kinds of angles. So when the information reaches the higher levels of the cortex, it's already processed so that we can detect contrast, we detect lines and edges, and we detect angles. And then they can combine those together to get even more complex kinds of objects and forms. So the visual system is designed to allow us to detect edges of things, which means we can tell figures from backgrounds, we can see objects, we can detect the things which we can then identify through our perception. Thank you.